A very big hello to my dear little children out there. Let's see if the class is officially live or not. Okay. Yes, it's live. Chalo. So those of you who are able to see me, hear me properly, give me a thumbs up right now. Am I visible? Am I audible? All good? And those of you who are seeing me for the first time, a very big hello, my dear children. So today we are here to have a complete revision of biology. And so I have taken MCQ questions and also the 2 marker, 3 marker and 5 marker questions. Hi Rishi. Okay. Hi Spectre. Okay. Giving me thumbs up, which means I am clearly visible and audible. Okay, so children, do you want me to start with MCQs or do you want me to start with the 5 marker questions out there? So there's a choice that I'm giving you guys. Hi Santosh. What is it? Is it MCQs? So the first chapter is sexual reproduction, flowering plants, and the MCQs are the ones that we did previously. And if at all you want me to directly go with 5 marker, 3 marker, I will be going ahead with that. Bhuvan, I am really great. Thanks for asking. Chalo. So someone says, ma'am, we have finished today's exam biology. How was it? Was it bad? Was it good? Okay. In that case, Spectre is saying 5 marker, Rishi is also saying 5 markers. So let's begin with 5 markers first, since we have already done the MCQs previously. Are we ready? So the first question that we have over here is this. Hi Ramesh. Picture related to pollination is given below. Will this pollination confirm fertilization? So insect is given, a plant is given. So what do you think? Will this particular pollination confirm fertilization or not? You never know. Pollination can happen, but fertilization totally depends on other factors as well. So we say no, we cannot confirm this. Now what are the floral rewards provided by the plants to the insects to revisit? So you have to talk about that. What are those? For example, nectar, for example, pollen grains, a place to lay their eggs. These are the 3 rewards or the 3 things that can be given by the plant to the insects. Now, mention the characteristics of flowers in the pollination rather than the floral rewards. So you have to talk about the the features of the flower. So over here you have the answer. First question's answer is what? No, pollination does not always confirm fertilization. You know, genetic incompatibility. For pollination is happening where fertilization is not happening. So obviously it is 2 different things. Pollination is what? The transfer. So you have to tell about the definition of pollination and then you tell about the definition of fertilization and say that they both are not linked to each other. Pollination happens and then fertilization can happen. Fine. Next thing, nectar. What are the things that you need? So as okay, I do not know if I'm pronouncing it correct, but Khadakalat says that nectar and pollen is the answer. Yes. So nectar, pollen grains as well as shelter. So you know yucca and moth. In that basically the yucca plant is giving a safe place for the moth to lay their eggs. So that is that. Now majority of insect pollinated flowers, now you talk about the features of the flowers. They are large, they are colorful, they are fragrant, they are rich in nectar, and sometimes they are small. If they are small, they are grouped together to look bigger so that the insects can see that easily. Okay, and animal attraction is by color and fragrance. And my dear children, there are certain plants which are not pollinated by the other insects out there, rather than they are pollinated by the flies, house flies and things like that. For that, the flower has to be foul smelling. So these are the things that you can definitely write down. This is how you get your 5 marks for this question. Okay, now going on to the next question, that is explain the structure of Megasporangium. So over here what you have to tell, first of all you have to make the diagram. If at all you're writing your answers without diagram, then you might lose your marks. So make sure you're making the diagram, and while you make the diagram, I'll help you with this. So look at this. Oh my god, just a second. I am clicking on the wrong thing again and again. So first thing, make the ovary. In the ovary, my dear children, you have the placenta. Placenta is connected to the ovule with the help of something which is known as funicle, and the point of attachment is known as hilum. And then the ovule which is there, it feels so cold, which is why it needs 2 sweater. So these are the 2 integuments that it has, and the opening is known as micropyle. The opening which is there, it is known as micropyle, and over here you have you have what? The cells of the nucellus. The cells of the nucellus, and one such cell, my dear children, which is known as megaspore mother cell, divides to form the functional megaspore and later the embryo sac. So you have to mention all of it, and this is the structure that you have to follow. Hi Asya. Okay, so please explain your answer properly one by one. That is, there is ovule, which is a small structure attached to the placenta by means of funicle, and the place of attachment is known as hilum. The ovule has two integuments, and there is micropylar end, and the opposite end is known as what? Chalazal end, and the cells which are there in the ovule, they are known as nucellus. So all these questions are, sorry, all these parts of the answer are really, really important. Make the diagram and then only explain. Okay, children, are you getting it? So still now, do you think that we should be going with the 5-marker question? Do you need explanation, or do you want me just to, what do you call, just the brief of it? So however you want, you can tell me. And as I said, I have made separate videos on it, whether 2-marker, 3-marker, one-shot video, everything is there. But I am here to take up your doubts as well as, what do you call, explain it better. Brief. Okay, let's go brief then. So picture of mature angiosperm embryo sac is given below. You have to tell the answers. So which cells, nuclei of the embryo sac, produce zygote, primary endosperm nucleus? So which cell do you think produces zygote? It is the egg cell. So over here you have the egg cell. The egg cell fuses with the male gamete to form the zygote. What is the ploidy of zygote? The ploidy of zygote is diploid. Hi Ellen. Okay, it is what? Diploid in nature. Now you are asked about endosperm, primary endosperm nucleus. Where do you think it is formed? So when the male gamete comes, there are two of them that come. So one fuses with the egg, female gamete, that is the egg, and the other one fuses with the two polar nuclei. So this is haploid. The sperm or the egg, the male gamete which is coming, that is haploid. The two polar nuclei, each of them are haploid. 3 nucleus come together. This is known as triple fusion. And at the end, what do you get? You get the primary endosperm nucleus. Now, what is the ploidy of it? It is triploid. Okay, it would be triploid in nature. So please tell that. Can you tell the fixed questions for bio midterm, man? So in this world, no one can tell you the fixed question apart from the people who made it. But definitely, I have made a separate video on short questions, which may probably come out there. So that video will be live really, really soon. Okay, Rishi? Okay. Fine. Now, what is the ploidy of antipodal cells and primary endosperm nucleus? So, you know, primary endosperm nucleus ploidy is 3N. And what is the ploidy of antipodal cells? All the cells except the central cell is haploid. Except the central cell, all of them are haploid in nature. The central cell, since it has 2 polar nuclei, it is diploid in nature. So it will be haploid. Now, and the primary endosperm, okay. Why the endosperm development precedes the embryo development? You should know that. Why do you think endosperm develops first and then comes your embryo? Because there should be someone to nourish the zygote to form the embryo, right? And that is done by the primary endosperm. So the endosperm acts as a nutritive tissue and provides essential nutrients to the developing embryo. Okay, fine. Early endosperm formation ensures a steady supply of food, which is why you have endosperm first and then the embryo. Now, going ahead with the next question out there, explain any 3 outbreeding devices in the flowering plants. What do you mean by outbreeding devices? The ones that does not allow self-pollination to happen. Okay, those are known as outbreeding devices. So in the plants, 3 ways to easily remember it. I'm not reading all of it. First one is the positioning of the anther and the stigma. Suppose the anther is here. Okay, the stigma is here. Do you think pollination can easily happen? No. So that is one thing, position of anther and the stigma. Second, different maturation time. Suppose the anther matures first and then the stigma. Obviously, they won't be receptive at the same time. That is how you can prevent self-pollination. Third one is known as self-incompatibility, which is a genetic mechanism where the plant itself, suppose the pollen grains are landing, but it won't allow what you call pollen tube formation to happen. Okay, that is the third thing. So that are the 3 outbreeding devices that you can do. Okay. Prakriti, are you, ma'am, in 10 minutes you finished 17 questions? No, dear, I didn't start off with MCQ. I directly came up to the 5-marker questions since children asked for that. Okay? So these are the extra things out there. You can read it. And there is formation of unisexual flowers. They also outbreeding devices. Why outbreeding? So suppose there is a plant who just has female flowers. Okay, obviously it will have pollination with another plant that will have male flowers. So their cross pollination is definitely going to happen. These type of plants are known as dioecious plants, where the sexes are separate in 2 different plants. Dioecious means sexes are separate in 2 different plants. So this is a female plant, this is a male plant. Example of this is papaya. You can get question from this as well. Okay. Fine. Now, going ahead with, okay, now this question is super important. Draw the LS of the grass embryo. Grass embryo, if at all grass embryo comes, just understand you're talking about monocot embryo. Okay, you have been just asked about the monocot embryo. Just look out for that. And this is the diagram that you have to make. Make sure you're making coleoptile as well as coleorhiza and all the other things properly. Ma'am, please MCQ practice. So how many of you say that we have to go ahead with MCQ practice? Then I'll go ahead back to MCQ practice. Anyway, these questions are there in the 2 marker, 3 marker, 5 marker session. But yes. Ganesh, if we watch this, can we get 65 plus? Yes, you can. Okay, now draw the label diagram of transverse section of young anther. You have to make the diagram, you have to mention everything. Epidermis, EMT, remember? Epidermis and then endothecium, middle layer as well as tapetum. So these are the things that you need to remember. Hi Sakina, I am doing good, thank you for asking. I hope you are also doing great. So Rishi is saying, teach me how to draw this diagram in question number 18. This diagram, okay, I can help you with that. So over here you make a structure like this, okay, and then make a small little hand like this, and then make a whale's tail. So it looks like whale's tail, right? And then over here there will be a small hand. There will be a small hand. Now over here we have to make one thing like this, and again one more thing like this. Okay, so this will be a shoot apex, this will be a coleoptile. That's how you can easily make it. And now my dear children, inside this you have to link all of it. So link this like this, and just mention that this is the shoot apex and this is what? This is your coleoptile. Okay, this is your radicle, and just a second, this is your radicle, and over here my dear children you would be having your root cap, and this would be your coleorhiza. So you need not make this, you don't have to make this. Okay, cool. This is how you can draw this. So I'll tell you, this particular diagram itself is not very much specified, which is why you can draw like this. It's not a problem. Yes, they asked this diagram and it's one of the most repeated diagrams out there, which is why I want you to practice. Okay, so whale's head and then 2 hand and whale's tail, and then inside you make like this, again you make one more, then join everything together, especially towards one side, make an extra root cap over here. The lower region is known as coleorhiza. Okay, 3, 2, 1 marker first, ma'am. So let me just finish this, then I will go ahead with that. Okay, now transverse section of anther, I hope you are done with that. And now my dear children, okay, describe the structure of dicot embryo with the help of label diagram. Again you have to make a dicot embryo. This is very simple. You make the embryonal axis like this, okay, and over here you have the plumule region. Now 2 hands, one hand like this, another hand also drooping like this. So this is your dicot embryo. So this is your plumule region, this is your radicle, and these are the 2 cotyledons that you are having. And over here, the lower region from the embryonal axis is known as hypocotyl, and the above region is known as epicotyl. The tip of epicotyl is known as plumule. Okay? Fine. Now, going ahead, write any 4 characteristics of flower pollinated by wind. So you have to talk about wind pollinated. I told you features are very, very important. So if it has to be wind, my dear children, Imam, e questions pakka kelkatara. Okay, yeah, keltara. So there are high chances that these questions can come because these are the repeated questions each year. Okay. Now, obviously, it's not like the exact question would be coming word by word. The concepts can be coming in. So please make sure you are, practicing it enough. Fine. Now, write the 4 characteristics of flower pollinated by wind. What are those? It should be the pollen grains, my dear children. Do you think it can be heavy? No, it has to be light so that it moves around with the wind. Now, the stamen which is there, it should be tall. Okay, it should be long in nature and well exposed. Why? Because the wind can carry the pollen grains then only. And the stigma which is there, Okay, the stigma which is there, which has to capture this pollen grains, there has to be really feathery, so that pollen grains can be easily captured. And usually the number of ovule in the ovary is just 1, because wind pollination may happen, but the chances of it to happen is also less. Even though in abiotic factors, this is one of the most common abiotic thing that is there, that is wind pollination. So these are the features that you have to remember, and also the example. The example of wind pollination, anyone? Can you guess? Can you guess the example of wind pollination? The plant that undergoes wind pollination. So we discussed this in the MCQ part. Any guesses? Any guesses? Vallisneria. Hi Shriyans. So it is not Vallisneria that shows wind pollination. Can you keep guessing? Good afternoon, Muskan. Children, the plant that shows wind pollination is an important question. It can come in the form of an MCQ. Yes, corncob and also what do you call it as? Corncob and also grasses. Two things. So corncob and grasses are the two examples. You need to remember this. This is in your NCRT, the last line of your NCRT. We solved the MCQ question as well. Okay, so corncob and grasses. Now what are false fruits? So usually it's the ovary that become a fruit. But suppose it's not just the ovary, something else is also helping in the formation of fruit. That fruit will be known as false fruit. For example, in the case of apple, now that diagram of apple can be given, which is given in your NCRT. So this is the thalamus, my dear children, onto which the ovary is resting. You know what? The thalamus also participates in the formation of fruit in the case of apple. So the thing that we bite, okay, that is the thalamus that had become a fruit. So this is what known as false fruit. So false fruit develop from any parts of the plant apart from the ovary, like the thalamus or the pedicel. Example, apple, cashew, as well as strawberry. Okay, cool. Next question. 2-marker question. Vinay, I think, was asking for the 2-marker question. So here it goes. So cells of the male gametophyte. What are the cells of the male gametophyte? First of all, you should know what are the male gametophyte. It is pollen grain. Now there are 2 cells in that. One is vegetative cell, other one is generative cell. The nucleus of the vegetative cell, the shape of it is asked. So when vegetative cell, it is irregularly shaped, whereas in the case of generative cell, it is elongated in shape. Okay, it is elongated in shape. Tara Devi, if you study this, can we score well? You can definitely score well. Okay. Can we go ahead? So next question, differentiate between geitonogamy and xenogamy. This is also one very important thing. Geitonogamy means what, my dear children? So over here, suppose this is a plant with 2 flowers. From here to here, pollination is happening. This is known as geitonogamy. So is it cross-pollination? Yes. But is it true? True cross-pollination? No. Okay, on the other hand, we have this particular plant and this particular plant. Suppose pollination is happening from here to here. Do you think pollination is going to happen? Say, do you think cross-pollination is happening? Yes. And is it true cross-pollination? Yes. Why? Because 2 different plants are involved. So the progeny that will be formed will be a combination of both of these plants. Okay? I hope that is clear. So geitonogamy, you have to mention like that. It is a transfer of pollen grains from the anther of one flower to the stigma of another flower of the same plant, and genetically similar to the parent plant. Why? Because, my dear children, it's from the same plant, everything is happening. And xenogamy, transfer of pollen grains from the anther to the stigma of a different plant. Genetically different from the parent plant, it brings out variations. You have to remember it. So out of this, who brings out variation? Xenogamy. You can get a statement. Geitonogamy is genetically like autogamy. Is it true or false? Geitonogamy is genetically like autogamy. Is it true or false? The answer is yes. Why? Because in autogamy also, from the same plant, suppose this is a flower, it has both the male and the female part. From here to here it's going. So genetically anyway, say the same pollen grain, same everything out there. Right? Same thing is happening over here also, just that the flowers are different. But the pollen grains are from the same sources. Okay? The pollen grains are from the same sources. Now going ahead, what is parthenocarpic? Give an example. So as I told you already. So over here, usually what happens? The ovule is there. Inside the ovule, you have the egg cell and it fuses with the male cell. That's how it forms a zygote, and now you call the ovule as seed. And then you call the ovary as what? Fruit. But over here, without all this happening, without the formation of seed, my dear children, or without fertilization, what is happening? The ovary is turning into a fruit. Why, ma'am? Because there are some hormones coming into action. So there are certain hormones which ripens the fruit or which ripens the ovary to form the fruit. Example, banana. Yes, Vinay, you are absolutely correct. What if both plants are genetically similar? There are very rare chances for that to happen. There will be at least some different variations, genetic variations out there, unless and until they are not clone plants, okay? So usually that is the very rare unless and people make it with the help of apomixis and things like that. Cool. In nature, it's very rare. Fine. And there will be small variations anyhow. So the example for parthenocarpy is banana. Always remember this. Can we make fruits parthenocarpic? Yes. Can we ripen the fruits? Yes. By injecting certain hormones out there. For example, auxin. Okay. So parthenocarpy can be induced as well. Always remember that. Now differentiate between pericarp and perisperm. What is pericarp, my dear children? Pericarp is the outer layer of the, the wall layer of the ovary. So the ovary which is there, suppose this is mango. In mango, this three of them come together to form something which is known as pericarp. The outermost layer that we peel off is known as exocarp. The thing that we eat is known as mesocarp. And the one that covers the seed is known as endocarp. Remember, the seed has its own covering. This is the extra shield which is there. Okay, this is the extra shield which is there. So this is known as pericarp, the wall of the ovary. Now what is perisperm? That is something very much different, right? So what is perisperm, my dear children? In the ovule, no? In the ovule, as I said, there are cells which are known as the cells of the nucellus. The cells of the nucellus, and one cell out of this divides and forms the embryo sac, where all the the fusion of the male and the female gamete and everything is happening. And usually this nucellus which is there, it goes off, it degenerates. Okay, but sometimes this particular nucellus can stay back. For what? For providing nourishment to the embryo. So this particular layer of the cells of the nucellus which stays back for nourishing the embryo is known as perisperm. And you find it in two types of plants, both of them starting from B, which is why you can give B square as a thing. Okay, it can be known as B square. So black pepper and beet is the answer for this. Okay. Ma'am, this video will help me get 50 plus. So all the questions in this video, if at all you practice, my dear child, I am pretty sure you're going to score really great marks, because obviously the concepts would be repeated. It's not like they will bring out some new concepts out there, right? So they will be following the old concept. And if at all you just want the questions and not the answers right over here, then there will be a separate video on it. Don't be worried. Okay, cool. So perisperm is there in black pepper and beet. No dear, it's Prakriti. In coconut you have endosperm. And my dear children, before I go anyway ahead, there is KC90, an online crash course that we have launched for the two exams, that is KCET as well as COMEDK, strictly online, and it is going to start from 1st of November. So if at all you want to make bookings, make sure you're calling at this number or probably going to the link in the description box. Okay, and if you have any queries, you can put that as well. Chalo. So going ahead to the next chapter, again I am asking you, do you want to solve the MCQs or do you want to go ahead with the 5 marker, 3 marker, 2 marker questions? So over here, I started off with the 5 marker, 2 marker, 3 marker questions because I've already done these questions in the live before. But if at all you want me to go with MCQs, I can definitely go with that. Okay, you want to go with the MCQs now? Fine. But my dear children, always understand, definitely we can do MCQs, but you have to solve. Okay. Preetham V, ma'am, I had, okay, you win. So I see 2 marker, 3 marker. Okay, so let's take vote. So those of you who are saying 2 marker, 3 marker, 5 marker, tell me right now that we want that. And those of you who want MCQs, tell me that. Let's see who are in bigger number, we'll solve that. MCQs I have already solved in the previous 2 lives, but if at all you want me to do it live again, I am ready to do that. If you want me to do 3 marker, 5 marker, I can do that as well. But if we take both, it might take a long time out there. So Ganesh is strictly, saying 2 marker, 3 marker and 5 marker. Okay. So we'll do one thing very quick to, to make both of you Guys, really happy. We'll do both, okay, but the pace would be really fast. So will you coordinate with me? If at all yes, then we can go ahead. Okay, if you're ready to coordinate, give me a thumbs up. We have to tell the answers fast. Okay, so the first question out there. Scrotum helps in maintaining the temperature of testis, which is A, lower than the normal body temperature. How much degree Celsius it is lower than the normal body temperature if it is in scrotum, my dear children? It is almost 2 to 2.5 degrees Celsius. So over here, and here you have got the options. Okay. Now you can exclude this and this. Next thing, each of the testes has about B compartments. How many compartments are there? Testicular lobules. There are almost 250 testicular lobules. So again, these two are having a tie-up out there. And now it measures C centimeter in length. What is the length of the testis? So always remember, over here the width of the testis is 2 to 3 centimeter and the length is 4 to 5 centimeter. So what would be your answer for this question? It would be B. Yes, Ganesh and yes, Prakriti and also Vinay, the answer would be B for this particular question. Okay? Correct. Now let's go ahead. During the embryonic development, embryo's heart forms at the end of. Ma'am, I had this particular MCQ. Variation of DNA is due to recombination, mutation, both A and B. Answer is both A and B. Okay. So yes. So you guys are saying that the embryo's heart formation takes place at the end of first month. That is the C option. You guys are absolutely correct if you think so. Always remember, when we die, we check for the heartbeat. Similarly, when someone is born, the first thing that we check is the heartbeat because it is formed after the first month. In the second month, you have limbs and digits. In the third month, you have most of the organs being formed and also what do you call the external genital organ, whether it determines whether it's male or female. Now, fourth month, skip Madi. Fifth month, you would be having the first movements of the baby and also hair on the head. Sixth month, there will be hair on the body. And there will be eyelids that will be separated and eyelashes also. Okay, so in the first month you have heart being formed. Now next thing, the vas deferens receives ducts from the seminal vesicle and opens into urethra as. Try this. Prakriti, Ganesh, you guys were correct. Let's look out for the next question out there. The vas deferens receives duct from the seminal vesicle and opens into urethra as what? Epididymis. So it is saying there is vas deferens and it gets secretions from the seminal vesicle. Together they open into what? Together they form what? That is the question. This is the third question. So Ganesh says the answer is B. What about others who are watching this video? My dear children, we want maximum one of you to tell the answers. Otherwise this class is going to be very boring. So Vinay Patil also says it's B. Is it B? Yes, it's B. It is known as ejaculatory duct. Okay, vas deferens, seminal vesicle opens into ejaculatory duct and this ejaculatory duct will be opening into urethra in the case of males. So this accessory ducts as well as accessory glands in the case of males is very important. Please make sure you go through it. So over here I have the picture. Vas deferens comes like this. You get the seminal vesicle. Both of them open into ejaculatory duct and they open together into the urethra. Okay. Next thing. The function of myometrium layer present in the uterine wall is to. What is the function of the myometrium layer? Myometrium is the muscular layer. Do you know that? It is what? The muscular layer. And what is its function? Since it is muscles, so it has to contract and relax. And that is going to help in the production of what do you call the ejection of the baby outside the mother's womb. So your answer for this question would be what? Exhibit strong uterine contraction during the process of parturition. Parturition is expulsion of baby outside the mother's womb, right? So Rishi and Rishi and Vinay, both of you are correct. The answer is B. Okay, endometrium is the innermost layer, it is granular layer, and perimetrium is the outermost layer. Fine. Going ahead with the next question. Sertoli cells are found in. Where do you find Sertoli cells? Ovaries and secretes progesterone, adrenal cortex and secretes adrenaline, seminiferous tubules and provides nutrition to germ cells, pancreas and secrete cholecystokinin. What is your answer for this question? Where do you find the Sertoli cells? So as far as I remember, you have the testicular lobules. Inside that you have the seminiferous testicular lobules. Inside that you have seminiferous tubules. Inside that you have the Sertoli cells and also the spermatogonia. The Sertoli cells are going to nourish the spermatogonia. That is the thing. So seminiferous tubules and provides nutrition to the germ cells. That is your answer. That is C. Yes, Rishi, Vinay, Prakriti, Ganesh, all of you are correct. Kudos to you. Okay. Now, what are the cells which are present in between the seminiferous tubules? They are known as Leydig cells, and what do they produce? They produce androgens. Okay, they will be producing androgens. Now, going ahead, the cells that secrete testicular hormones are, I just told you right now, the cells that secrete the testicular hormones, what do you call them? Testicular hormones means the male sex hormones, androgens. Can you tell me the answer like 7, A, B, C, D? That will be more easy for me to understand whether you're telling the correct answer or not. The cells that really secrete testicular hormones are what? Vinay says the answer is B. Okay, anyone to challenge Vinay and say it's wrong? Ganesh says the answer is V or B. There's no option like that. Okay, he said B. Okay, now it's fine. What about the others? So 19 people watching this video, chumma, they're not even answering. Ayye. Come on children, answer. Yes, Prakriti says it's B. Okay, so everyone who said it's B, you are absolutely correct. It is Leydig cells and we just discussed about them. So this is seminiferous tubule where you have the Sertoli cells and the spermatogonia, and in between the seminiferous tubules you have the Leydig cells which will be releasing the androgens or the male sex hormones, especially testosterone. Now next, seminal plasma in human males is rich in. Seminal plasma in human males is rich in what? What is seminal plasma? So the accessory glands of the males, there are 3 of them. Seminal vesicle, prostate gland and bulbourethral gland. 3 of them secrete certain things for the sperm, for their nourishment, for helping them in some or the other ways. Okay, now you are being asked what the secretions, what does it contain? What does it contain? What is your answer for this question? Prakriti says 8A is the answer. Vinay also says it's A. What about others? Come on, dear children. Try Madi. A. Let's go for A. Fructose and calcium. What releases fructose? Ganesh also says it's A. Good boy. So fructose, my dear children, is being released by this guy, that is seminal vesicle. Remember, it's not glucose, it is fructose, and fructose provides energy for the sperm's movement. And calcium is produced on the other hand by prostate gland. And always remember, the question can be seminal plasma releases enzymes as well. Has enzymes as well. The answer would be correct. Why? Because prostate gland releases 2 things. One is calcium and the other thing is enzymes. So don't get it wrong. And bulbourethral gland, it helps in the lubrication of the penis. So there are 3 things out there and you have to remember the names. Together the secretion is known as seminal plasma, and with sperm now you will call this as semen. Okay? Now going ahead with the next question. Statement 1. The process of release of sperms from the seminiferous tubules is known as spermatogenesis. Do you think the process of release of sperms from the seminiferous tubules is known as spermatogenesis? Do you think so? And statement 2 says the spermatids are transformed into spermatozoa by the process called spermiogenesis. What is the answer for this particular question? Question number 9. So those of you who are just watching the video and not answering the question, let me give you a friendly reminder that, my dear children, we learn things better when we do mistakes. That does not mean that you have to do mistake, but if at all you're worried that I might probably be wrong while giving the answers, it's a, it's a very negative thing to think of. Give your answer, see if it's wrong or correct, and then if it's wrong, correct it. That's all. Simple. Chalo, that's the spirit. So we have Prithvi as well as Vinay, Rishi, everyone telling the answers. And over here, so you are saying D is the answer and someone is saying, Bhuvan is saying A is the answer. Okay, let's see what is the answer. So the process of release of sperms from the seminiferous tubules is known as spermatogenesis. Wrong. What is spermatogenesis? The formation of sperms, the whole process of formation of sperms. The release of sperms from the seminiferous tubules is known as spermiation, aviation, you can remember like that, spermiation. Okay. Now next thing, the spermatids are transformed into spermatozoa. Is it? Yes. Spermatids, they are like kids, they are immature, they are round, but we want long sperms. So this transformation of kids to spermatozoa, of spermatids to spermatozoa, is actually known as spermiogenesis. Okay, and remember, it needs high level of FSH and this FSH will go act on Sertoli cells, and the Sertoli cells will produce certain factors which says that testosterone, you have to be here so that this particular process can smoothly happen. So if at all spermiogenesis come, just related to FSH, Sertoli cells, and as well as all those things I have written out there. Okay, fine. So your answer for this question is A is wrong, B is correct. So technically speaking, D is the answer. Okay, statement 1 is incorrect, but statement 2 is correct. So those of you who got the wrong answer, no issues at all. I hope now you know the answer, right? Now 10th question, is spermatogenesis if FSH hormone is not secreted from the anterior pituitary gland? Which stage is affected? I just told you right now. In spermatogenesis, if FSH hormone is not secreted, what will happen? Simple. Okay, what will be formed, what will not be formed? What do you think is the answer for this question? So this is how we relate and learn, and this particular picture is really important. Okay, come on dear children, tell me the answer. 10 A, B, C, D. Alina, you didn't miss anything dear. Okay, Alina is here saying the answer is C. Which stage is affected? That is the question. Prakriti says it's B, Ganesh says it's C. Okay, we have a tie out there with B and C. The answer for this question is B. I just told you what happens, my dear children. So over here, spermatids which are there, they convert into, they're kids, right? So they convert into the mature form which is known as spermatozoa. And this process is known as spermiogenesis. Over here, the FSH, when they act on the Sertoli cells, the Sertoli cells are going to release some factors. Now you might think, why do you need these factors, ma'am? So these factors are the one that will be telling the testosterone, the male sex hormones, to stay here itself so that this process can happen smoothly. Now if these factors are not there, testosterone would not be here in high amount and this process would not be happening. So formation of spermatozoa would be affected. So which is why our answer for this question would be B. Always remember, FSH, pituitary gland, releases two things. One is LH, luteinizing hormone, another one is follicle stimulating hormone. LH acts on Leydig cells, LL, and the Leydig cells are the one that produces what? Male sex hormones. Now over here, FSH acts on Sertoli cells and they release some factors. The male sex hormones will move into seminiferous tubules where all these guys are there, and they just want to move around, they want to move away from these guys. But Sertoli cell says that you have to be here, don't go anywhere because I need you for this particular process to happen. Okay, actually testosterone is needed for the whole process, but majorly for this particular process according to your NCERT. Okay, so next time don't do this mistake, give this as the answer. Next question, easy question. To produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? To produce 2000 sperm and 400 ova, how many spermatogonia and oogonia are required? Over here, always remember, from one spermatogonia you get 4 sperms, and from one oogonia you get only 1 ova. Okay, from 1 you get 4 here, but over here from 1 you get only 1. Ganesh says, I have enrolled KC90, I'll get below. Superb, Ganesh. That's so good. Wishing you all the best. I hope we help you in the preparation really well. Okay, 11B is what you say is the answer for this question. 500 spermatogonia and 400 oogonia. Is that correct? Well, I'll say it's correct. Why? Because, my dear children, 2000 sperms. Okay, divided by 4. Because in every time you get 4 sperms, right? So 2000 divided by 4. So we'll get to know how many spermatogonia needed. So almost 500 spermatogonia needed. 500 of them divide, each of them produce 4 sperms, which is why at the end you have 2000 sperms. Now what about the oogonia? There are 400 oogonia, 400 divided by 1, it is 400 only, no. 400 ova is there, you will get 400 oogonia also. So your answer for this question would be B. Yes Rishi, yes Vinay. Okay, so your answer is B, don't get this question wrong. Now going ahead with the next question out there, which among the following has 23 chromosomes? Try it out. Everyone who is watching this video has to try this question. Very simple. Which among the following has 23, just 23 chromosomes? Not beyond that. 23 means haploid number of chromosome. Usually we have 46 number of chromosome and the half of it is 23. So you are being asked who has 23 chromosomes, just 23 chromosomes. Alina says the answer is C. Let's see if Alina is correct or not. Prakriti also says it's C. Okay. Prithvi says it's D and Rishi says it's B. Okay, so you guys are giving me MCQ question over here, A, B, C, D. So my dear children, 23 chromosomes over here if you see. Okay. Spermatogenesis is there. It undergoes differentiation. It's not like it undergoes any divisions. Here it is wrong. Okay, don't think it's mitotic division. It's not mitotic division. It undergoes differentiation and it forms primary spermatocyte. Right? The primary spermatocyte is diploid in nature. Now it will undergo meiosis, and once it undergoes meiosis 1, the number of chromosomes are going to reduce into half. So in secondary spermatocyte you will have haploid. In primary it is going to be diploid itself. From secondary all of them are going to be haploid in nature. The second meiosis happens, but here reduction does not happen. Here the number of chromosomes remains to be the same. So in this question you are asked, which is that stage where you have 23 number of chromosome? It is what? Secondary oocyte. So as of now we discussed about spermatocyte, but the same thing happens with oocyte also. So if you see over here, you have the oogonium. Come on, just a second. Okay, here you have the oogonium. Here you have the primary oocyte with 2N. It undergoes meiosis. You have first polar body and the secondary oocyte. Here it's when it gets half, not before. And now it remains the same. N remains to be N in the ovum as well. So your answer for this question is secondary oocyte, not primary oocyte. Always remember. Okay. Now I'll ask you a question and you have to give me the answer. Since I remember this, I'm asking you right away. So your answer for this question, following has 23 chromosomes, would be what? Would be secondary oocyte. Now the question to you guys is, which among them would be having 3.3 into 10 to the power 9 content of DNA? 3.3 into 10 to the power 9 content of DNA. Your options are A, B, C, D. You can give this as 12 homework and then tell the answer. What's the answer for 12 homework? Which of these cells has a content 3.3 into 10 to the power 9? Now my dear children, I had explained this to you in the yesterday's class, if at all you remember. What is the normal diploid content of our, what do you call, DNA? It's 6.6 into 10 to the power 9. Always remember. Okay, it is 6.6 into 10 to the power 9. Over here, it is 3.3. So this is diploid. That means 46 number of chromosomes. If it's there, it's like this. But haploid happens over here. And it will be like this. So your answer remains to be the same. It is secondary oocyte. Okay, it would be secondary oocyte. Remember this, very important. It can come in molecular basis of inheritance. So don't get it wrong there. Okay, Sirisha Sri. Yes, C is the answer. Okay, do not forget it. Going ahead with the next question. Identify the incorrect pair with respect to menstrual cycle. Which one is incorrect, my dear child? So, over here, formation of corpus luteum occurs from the 15th to 28th day. Secretion of progesterone occurs from the 15th to 28th day. Regeneration of endometrium 5th to 13th day. Disintegration of endometrium 5th to 13th day. What is your answer for this question? Come on. Let's go. 13, the answer is D. Okay, cool. So Prakriti says it's D and Alina says it's C. Let's see. Sirisha also says it's D. Kudos. Vinay D. Okay. I'm seeing more people going on coding for D. So yes, the answer is D. Listen to me carefully. From the 1st to 5th day, the endometrium, imagine, okay, the endometrium, it falls down. 1st to 5th day, that's when the menstruation is happening. And now after falling down, what happens? The follicles are going to regenerate under the influence of FSH and LH. Under the influence of FSH and LH, the follicles are generating now. When they generate, my dear children, they will be releasing some other hormones which are known as estrogen. And because of estrogen, the endometrium that just fell down slowly regenerates. So regeneration of endometrium occurs from the 5th to 13th day. And over here it is written disintegration of endometrium occurs. So that is wrong. Regeneration will be happening. Okay. Always remember. Cool. Yes. Alina, it is repeated. Yes. I did the MCQs on Saturday. I told you, right? So on Saturday and Monday, I did MCQs. And here I have included the 5 marker, 2 marker, 3 marker. But since children asked for MCQs, which is why I am coming back over here. But we will be discussing the 5 marker, 2 marker and 3 marker as well. Okay? So your answer for this question is D. Disintegration of endometrium does not happen over here. It's regeneration that happens over here. Okay? Now the next question. Which of the following statement is true? LH surge induces menstruation. Do you think LH surge induces menstruation? Okay. Regression of corpus luteum increases progesterone. Diploid egg is formed after second meiotic division. Embryo differentiates from the inner mass of blastocyst. Yes, Alina, please, you can go. Okay, no issues. So those of you who have attended the previous class, you may go, but if you want the 2 marker, 3 marker, you can stay back as well. Okay? Which of the following statement is true, my dear children? So LH surge induces menstruation is wrong. Why? Because LH, when it surges, my dear children, it induces ovulation. The what do you call the secondary oocyte that is being released. So it's not menstruation but ovulation. Regression of corpus luteum increases progesterone. No. You know what? Corpus luteum is the one that releases progesterone. So if corpus luteum is not there, do you think progesterone will be there? No. So regression of corpus luteum decreases progesterone would be the correct answer. Now diploid egg is formed after second meiotic division. Do you think anything is diploid in egg? No, it is haploid. Okay, it is going to be haploid ovum that is going to be formed after the second meiotic division. Always remember. Embryo differentiates from the inner cell mass of blastocyst. That is also correct because that is correct. That is the only statement which is correct because in the blastocyst you have the trophoblast which forms the placenta and there is inner cell mass, the cells which are inside. They actually form they differentiate to form the actual embryo out there. So this is the only statement which is correct and let me see who all gave me the correct answer. So Sirisha and Ganesh as well as Vinay gave me the correct answer for this question. Now, the next question. Match the following items given in column 1 with those in column 2 and select the correct options given below. Proliferative phase. What do you call this phase as? What is the other name for this particular phase? Secretory phase. What is the other name for this particular phase? Menstruation. What do you mean by that? So, we all know menstruation means what? Breakdown of the endometrium lining, the innermost lining. Right? So, C goes with 1. With that, can we find the answer? No, because there are 2 options like that. So, let's cut out these options. This is known as elimination method. So, Prakriti says the answer for this question is C. What about Sirisha? Sirisha is saying it's C only. Okay. Now, my dear children, always remember. So, menstruation happened. After that, as I said, follicles are going to generate. Now, follicles are generating, parallelly they are releasing estrogen that generates the endometrium. So, this is known as proliferative phase as well. Follicular phase as well as proliferative phase. Now, once the follicles are matured enough and the regeneration has happened, my dear children, what will happen? There will be ovulation. There will be LH surge, which is why ovulation would happen, egg would be released. Now, whatever is left behind in the Graafian follicle, that is going to become the corpus luteum. Now we enter the next phase which is known as luteal phase. And the corpus luteum is going to release 2 hormones. One is estrogen and the other one is progesterone. And progesterone is released in more amount. Which is why what will happen, the endometrium which was regenerated, it will become much more thicker and it will start secreting certain things as well. Which is going to help the embryo in development if at all embryo formation takes place. So it is known as secretory phase as well. So B goes with 3. Okay, which is why your answer for this question is C. A goes with 2 and B goes with 3 and C goes with 1. Cool? Fine? Next, going ahead with the next question. The secondary oocyte when released is covered by non-cellular layer. What is the name of this layer? Let me read out the name of the children who gave me the answer. Sirisha, Ganesh, Vinay, Prakriti, all of you doing super good. Okay? What about the other children out there? Chalo, answer. You will answer, you will remember things better. Okay, so Ganesh is saying the answer for this is C. Is it C? So you know what? It is the egg which is there, it is not released in the form of ova. Okay, it is released in the form of secondary oocyte. The secondary oocyte is not able to finish its meiotic division. It finishes it when fertilization is about to happen. Okay, so secondary oocyte, when it is released, it feels so sad and it needs protection, which is why it secretes one very important thing, which is known as zona pellucida. And zona pellucida, it is known as non-cellular or acellular layer. It does not have any cells around it. Okay, apart from it, they are also layered up by corona radiata, but these cells are made from the granulosa cells in the follicle. Okay, so your answer for this question would be C. Manasa is here, Rishi is back again. So answer is C, my dear children. And now we are getting into the 5-marker questions. Ready? So for some time we would not be having the MCQ. Just bear with me, listen to me carefully if at all you think you need marks from the 5-marker, 3-marker session. Okay, so draw a label diagrammatic sectional view of human female reproductive system. This picture is super important. Go through it very well. You have the ovary, the fallopian tube. Here you have the fimbriae. Then you have the infundibulum. Then you have the ampulla. Then you have the isthmus. This region is known as uterine fundus. This region is known as cervix. And over here you have the vagina. Okay, if at all you want me to draw the diagram and show you how to make things easy, you can let me know that. Otherwise, we'll just move ahead. So this came in the model question paper the previous year. So I'll just move ahead. I think we can practice diagrams later. Now the next diagram that comes usually is mammary glands diagram. Again, super important. You can remember it like this. Dal fell on mat. Dal fell on mat. So M stands for mammary lobes. Okay, M stands for mammary lobes. Mammary lobes would be having so many alveoli, and the alveoli would be opening into mammary tubules. The mammary tubules would be just a second. The mammary tubules would be tubules would be opening into ducts. The ducts would be opening into ampulla, and the ampulla would be opening into lactiferous ducts. So dal fell on mat, but mat has to come first. Okay? Fine. Rishi, you're talking about mammary gland diagram? Okay, so this is one very important thing that you need to draw and label as well. Draw properly, and remember A is repeated twice. So A, ampulla has L, right? So A after L is ampulla. And over here alveoli also has an L, but ampulla ends with an L. So you can remember like that. Okay? So if you're talking about the structure of mammary gland, it's quite easy. So over here you have to start off first of all by giving a direction of this particular thing. And now towards the side you have to make the fat. So whatever fat globules are there, you make that on either sides. Okay, and then make the lobes. So lobes can be made like this. Inside the lobe you have alveoli. So this is not my side. Okay, so the alveoli which is there, it is going to open. Similarly, you have to make another. Why is the pen getting away again and again? Okay. So the other alveoli could be made. They are opening into tubules. These are the tubules that you see over here. Okay. Now the tubules would be joining together to form what? The duct. Now you make a similar kind of thing here also and make 2 ducts. Okay. My pen is not penning. Okay. So over here you make another duct. Both the ducts would be joining to form what? An ampulla. And now again from here also you get another, my dear child, oh my God. From here also you get another ampulla, and the ampulla would be joining together to form the lactiferous duct. So that is how if you remember this particular thing, it will be very easy for you to draw that way. Okay, so each lobe you have to make this, and at the back side you have to make the ribs. So you can make the ribs like this. Okay, and mention the nipple. So you have to, lactiferous ducts which are there, they come like this, lots of them from each of them. Okay, and this particular region would be known as the nipple, and the outside layer which is there, you can make it double and you can show that this is areola. That is a tissue out there. Okay, so basic idea you will get only if you know this particular thing. And mention this to be the muscles. Here you can mention the muscles between the ribs. General thing, you don't have to make like Picasso. Okay, remember you would have very limited time to draw. Now the sectional view of male reproductive system is the next thing that usually comes up, and over here sectional view is actually this, but mostly the examiner wants this particular thing. I do not know why they put it as sectional view, but this is the picture that is usually being asked. If at all you think there is a chance for confusion out there, please talk to the teacher who teaches you biology at school. Okay, Rishi, I am very bad at drawing, but I'll try my best. Yes, definitely. So they are not expecting you to draw a very pro version of things. It's just that everything should be visible and it should be labeled properly. So hazard labeling, put it 1, 2, 3, 4, 5, 6, it will be more than enough. Okay. So this is the male reproductive system sectional view. Please draw that as well. Now going ahead to the next question. Write the schematic representation of spermatogenesis. When it is asked, you have to talk about spermatogonia. That spermatogonia is there. They will divide, undergo mitosis to form multiple number of them. And then one such thing will mature into what? Primary spermatocyte. The primary spermatocyte, which is diploid in nature, undergoes the first meiotic division, and then it will be forming what? Secondary spermatocyte. This will again undergo second meiotic division to form what? Spermatids. So these are the spermatids. And then it forms, the spermatozoa. This particular transition is known as what? Spermiogenesis. All the small things are super important to do that. Now write the two events that occur in the ovary and the uterus during the follicular phase of the menstrual cycle. All the phases of the menstrual cycle are super important, my dear children. So over here you are asked about the ovary as well as the uterus. What happens during the follicular phase? So as I told, in the follicular phase, the follicles are developing, they are maturing. And under the influence of what? Under the influence of FSH as well as LH. So please mention that. Under the influence of FSH and LH, the follicles would be developing into mature Graafian follicle, right? And when they do, what will happen? They will release some amount of estrogen, and this estrogen is going to help in the formation of endometrium. So in the uterus, what is happening? The endometrium is going to be formed in the uterus. In the ovary, what is happening? The follicles are generating and they are releasing hormones as well. So all the small details, super important. Okay, write in points and be calm while you write the answer. Okay, next. 3 marker. Parturition is induced by complex neuroendocrine mechanism. This is a very important question. Okay. Neuroendocrine mechanism means nervous system is involved, and also endocrine means hormonal system is also involved in this particular process. So how do you explain that? So first of all, remember, suppose there is a baby inside the mother's womb. The baby as well as the placenta, fetus as well as the placenta, both of them are going to give one fetal ejection reflex, the first reflex out there. By that I mean they are going to release a set of hormone that is known as prostaglandin. You do not have to remember the name. So this hormone, you know what it does? It causes mild uterine contraction. It causes contraction in the uterus. What all things? The fetus as well as the placenta, they release some hormones that causes mild uterine contractions, and now when the contraction happens, the baby will slightly move down. When it moves down, my dear children, the lower region of the uterus, you know what you call this? You call this as cervix. The cervix would be having certain receptors, stretch receptors. Okay, they become active. When they become active, they are going to send signals. Now this signal is what? A nervous signal. It is going to send a signal to what? Mother's pituitary gland, not babies. It is mother's pituitary gland. What pituitary? Posterior pituitary. We have two types of lobes, anterior lobe and posterior lobe. Anterior lobe gives the FSH and LH. Over here, the posterior lobe is going to release one very important hormone that is known as oxytocin. This is a hormone. So nervous thing giving what? A release signal to release a hormone, that is oxytocin. Now this oxytocin which is there, it will come here through blood towards the uterus, and here we have the myometrium, the muscular layer. Remember, the myometrium has certain receptors for oxytocin. So the oxytocin comes and binds over here, and when it does, again there will be contraction, relaxation. The baby will try to move down. Again, this loop goes on. This is known as positive feedback loop. So you have to explain point by point as I have written over here. But start with the fetal ejection reflex, which is given by both the placenta as well as fetus. It is usually asked. So who gives the signals for parturition? It is placenta as well as fetus, both of them. Okay, so fetus releases certain hormones, placenta converts into something else. So this is the thing. Okay, Rishi is asking, after this chapter, can we do molecular basis 5 markers? Yes, we can. So when the fetus is full term, its head pushes against the cervix, activating the stretch receptors, nervous signals goes to the brain, oxytocin release happen, and then strong uterine contraction, and there is positive feedback loop. This is a very important question. Okay. Next, in the figure given below, part A and part B shows the level of hormones, this other thing, in the menstrual cycle. Study the figure and answer the question that follows. You have to tell that. So name the organs, glands which secrete the hormones represented in labeled part A and B. So as I said, A is what? Pituitary gland. It is pituitary gland. It is releasing FSH and LH. What is B, my dear children? B is ovary. And what is ovary releasing? Estrogen and progesterone. Okay. Now, state the role of hormones secreted from part B on the uterus of human female. So, in the part B, there are 2 hormones being released. What is its role is being asked. What is the role of estrogen? As I told you, write like that. In proliferative phase or in follicular phase, estrogen alone will help in regeneration of endometrium. In the luteal phase, progesterone comes into action and makes the endometrium even more thicker and also makes the endometrium release some things, okay, release some secretions such that if at all fertilization happens, it can nourish the embryo. So have that thing in your head. If at all you are not writing all these things, it can lead to what you call a wastage of marks out there. So pituitary gland and ovary I have written. Estrogen proliferative phase stimulates the endometrial growth and thickening, prepares the uterus for implantation. Progesterone, it is the major thing. Okay, it is a major hormone. Stabilizes and maintains the thickened endometrium and enhances the glandular secretion. If fertilization does not occur, both the hormones level drops down and menstruation happens. Okay, so explain like that point to point. Even if you do not write this, it's totally fine. Just write this and mention, rather than putting in what you call bracket, explain. It increases in the proliferative phase and all those things. Fine? Done? Okay, now next. During the pregnancy, the level of certain hormones are increased several folds in the maternal blood. Name the hormones and write their importance. You know what? And by the way, Prakriti, good, and all the other children who are watching this video, I hope you're understanding. If at all you're understanding, do not forget to subscribe to the channel and also click on the like button. It's too hot up here. Okay, so the level of hormones are increased in several folds in the blood. So first of all, there are hormones which are only released during pregnancy, the placental hormones. So placenta is formed only during pregnancy, which is why it is known as, what do you call this as? Temporary endocrine gland. Endocrine is something that releases hormones. So you have the placental hormones, which are human chorionic gonadotropin, progesterone, estrogen, and human placental lactogen. So you might think, ma'am, these things are released by ovary also. Yes, but placenta, when it comes into action, they are the one that takes up the function. Now, the hormone that is released by ovary is this one. So ovary during the later phase of pregnancy, okay, it releases relaxin. What does it do? As the name suggests, it relaxes the, what do you call, ligaments, such that the baby can easily move out, but only during the later phase of pregnancy. Now this question is twisted. Why? Because they are not asking you to write all these things. Instead, during pregnancy, the level of hormones are increased in several folds in the maternal blood. That means there are certain hormones which are there, but in pregnancy they increase. What are those hormones? So you have to talk about 3 hormones. One is cortisol, other one is thyroxine, and the other one is what? Thyroxine, cortisol, and there is one more hormone which is prolactin. So these are the hormones that increases several folds. So if question comes and you are confused with the answer, write them, tell that they help in the metabolism of the mother as well as the baby and all these things, and then mention that placenta also releases some hormones. Ovary also releases some hormones. Extra thing that you can give. Okay, but make sure you're writing all this, otherwise you might lose marks. So these are the things, and you can talk about where are they released from, what do they do. You do not have to write in detail because in NCERT it's not in detail. So prolactin, lactation, it helps in the mammary gland development, so you can remember like that. Thyroxine and cortisol, you can just remember it's for metabolism and things. Cool? Works? Yes, Prakriti. Yes. Chalo, next. Principles of inheritance and variation. So Rishi was saying let's start with molecular base of inheritance. So I'll directly go to that chapter first and then we will come to this thing. So my God, oh my God, oh my God. Yes, we are here. So again, when we are in molecular base of inheritance, the first thing that I want to ask you is, do you want the MCQs first or the 5-marker, 3-marker things? Choose first. MCQs first or the 5-marker. MCQs have been done in the previous class on Saturday as well as Monday. So depending on what you want, I will begin. 5-marker. Okay. Chalo. Fine. 5-marker is over here. Where is 5-marker? Okay. List down the salient features. Okay, this is the first thing. List down the salient features of the double helix model of DNA. So you have to talk about the DNA. What is the structure of DNA? I hope you guys know that there is what? Sugar, pentose sugar. So that means it has 5 carbon. 1, 2, 3, 4 and the 5th carbon is outside. On the first carbon, it is bonded to nitrogenous base. On the 5th carbon, it is bonded to phosphate group. Okay, with the help of some oxygen. What do you call this bond? You call this phosphoester bond. You have to remember the name of the bond, phosphoester bond. What do you call this bond? You call this N-glycosidic linkage. N-glycosidic linkage. Okay, now this is what? One nucleotide. Similarly, there will be another nucleotide. So the top, suppose over here you have another nucleotide. Now the 3rd carbon of this nucleotide and the 5th carbon of this nucleotide is joined to the same phosphate group. So here also there is phosphoester bond. Here also there is phosphoester bond. If at all you are being asked, what's the bond that is between 3 and 5 carbon of 2 nucleotides? It is phosphodiester. Because why? 2 times ester bond is there. So it will be phosphodiester bond. And this is N-glycosidic linkage. Now we have to talk about other features. So what are the other features over here? First, DNA is double helix, okay, with 2 antiparallel polynucleotides. For example, suppose one is over here where you have the 3' end free, then opposite end will be 5' end. Similarly, here it will be 5', here it will be 3'. Why? Because the nitrogenous bases has to be facing each other. That's why they are ulta. So they are anti-parallel, not parallel. If at all it was parallel, it would have been 3 and 5 here also. But it is anti-parallel in nature. Always remember that. Now chains are coiled in right-handed manner. It's coiled in right-handed manner. The phosphodiester bond links between the 5 and the 3 direction on one strand and 3 to 5 on the other. Hydrogen bonds between the nitrogen bases. So there are 4 nitrogenous bases. Okay, 2 of them are known as, okay, just a second. 2 of them are known as purines and 2 of them are known as pyrimidines. So purines are what? Ag. I don't know why. Okay, just a second. A and G, and pyrimidines are known as T and C, thymine and cytosine. Always remember, in DNA you have thymine, but in the case of RNA you have something else which is known as uracil. Okay, fine. So you have to talk about them. A and T are bonded to each other. T for 2 bonds. Okay, G and C, since they look very similar, they have tightly bonds. So there are 3 hydrogen bonds. So mentioned like that, and it follows the Chargaff's rule where purines is equals to pyrimidines. That means A and G is equals to T and C. Why? Whatever number of A is there, that means number of T will be there. Whatever number of G is there, that means number of C will also be there. Now one complete turn of the helix. This is super important. You can get questions on the basis of it. So please listen to me carefully. This, okay, just a second. This whole thing is one particular helix, okay, and over here you have 10 base pairs. In one helix, how many base pairs? 10 base pairs. What is the total length? It is 34 Armstrong. It would be 34 Armstrong. So 34, and if at all you are asked what is the distance between all of them, so 10 of them are there, so 10 divided by 34 Armstrong, it will be 3.4 Armstrong. If at all your question is in nanometers, you have to talk like that. Okay, so 34 Armstrongs means 3.4 nanometer. So 3.4 Armstrong means 0.34 nanometer. That is the distance between them. So this would be 3.4 nm. And the distance between each of them would be 0.34 nm. You can get questions on the basis of this. Make sure you read everything properly. And now pentose sugar and phosphate forms the backbone of the DNA. Okay, the sugar and the phosphate which are there, they form the backbone. Okay. Now the next thing out there, describe the steps involved in the DNA fingerprinting method. So this question we will do a bit later because it might need explanation. Okay. This question is there. Answer the following. Construct a complete transcription unit with the promoter and terminator on the basis of the hypothetical template strand given below. Write the RNA strand transcribed from the above transcription unit along with the polarity. So this question is very simple. So this strand is given to you and now you do not know, ma'am, what does it stand for, 3' or 5'? How do I get to know this? How do I get? So here it is clearly mentioned. Construct a complete transcription unit with the hypothetical template strand given below. So this is a template strand. And template strand, my dear children, it will be in 3' to 5' direction. Okay, it is going to be in 3' to 5' direction. So from here to here, from 5, the new strand will be formed from 5 to 3 direction. Okay. Now you have to make a transcription unit. How do you make it? So look at this particular picture. look at this particular picture. You have to make an opposite strand. This would be the coding strand, and this particular region as a whole will be known as promoter region. On the hand, this will be known as the terminator region. Okay, promoter region, terminator region. Now this whole thing is a transcription unit. Mention this to be the coding strand as well. This is the coding strand, and that would be the template strand. Okay, here it is 3' to 5'. The coding strand will be from 5' to 3'. Okay, always remember on the 5' side of the coding strand you have the promoter region. Now you are asked this very important question, that is, can you tell the RNA transcribed from the above transcription unit? So anyway you have made the transcription unit right over here, the coding strand. Now my dear children, if you replace the T with U, because in RNA you do not have thymine, you have uracil. If you replace the T with U, we will be able to get what? The new strand of mRNA as well. You do not have to write it, you do not have to make it again. Just replace the T with U and you get the new strand, but always make sure it is from 5' to 3' direction. Otherwise, you're going to lose your marks out there. Okay? Cool? Fine. So next is the list the salient features of human genome. So before I go ahead, tell me, are you understanding? Or is it going like this? If at all you are understanding, give me a thumbs up right now. How do I understand that you are understanding or not? Give me a thumbs up if at all you are listening, you are understanding. Okay, Prakriti says, IVNR, ऊपर से जा रहा है. अरे ऐसा नहीं जाना है. We do not want it to go like that. So whenever you have a doubt, ask, okay? And do not feel demotivated at all. Ask the question, and my dear children, don't worry if at all you are not able to understand through this live session. So this is particularly aimed for those children who have at least certain background of studying these chapters or topics out there, because at this particular point of time when you have so much crunch time, you cannot go on explaining everything, right? But I have made separate video of 1 shot, of the 2 marker, 3 marker, 5 marker, where I explained in detail. You can go watch it as well. Okay? IAS dreamer Abhi, you will get out of out. I'm telling you, okay? So don't worry, just read. So you have to talk about the human genome, the salient features of the human genome. This is not something that I have to explain. This is something that you would have to dig into your head. There are points that you would have to remember. So the human genome contains of what? 3.64.7 million base pairs. So million term you can remember because over here million is being repeated, not billion. So 3,164.7 million base pairs are there. And you know what? The average gene size is usually 3,000 bases. A gene contains 3,000 bases. But there is a protein for dystrophin that has 2.4 million bases. Okay, how many million bases? 2.4 million bases. Now, the total number of genes which is there, it is estimated to be 30,000. You don't have to remember the other data if at all you think it is too much for you. So it was estimated to be 80 to 1,40,000, but it is just 30,000. And now, the functions of over 50% of the discovered genes are not known. We do not know that. So 30,000 genes are there, but out of that 50% we do not know what is their function. Now less than 2% of the genome codes for protein. Only less than 2% actually codes for protein. What are the other things doing then? They are just repeated sequences. They are repeating largely. They are not coding for any protein out there. Ma'am, in that case, what is the use of this repeated sequence? Is there any use? So basically, it can be repeated in different numbers in different people. So it can be repeated 8 times in my body, in my cell, in yours it can be 10, and depending on that we can be different. Okay, so that is really important for understanding the basic differences between us. Okay, and the evolution as well. So an organism and me might share the same DNA but in different numbers. So that's how we can relate. Oh, probably this organism might have evolved from me or whatever things, or you or any organism out there. Okay, now what are the important things that you have to remember? Chromosome number 1 has the most number of genes. Okay, it is 2968. It can be an MCQ question. And now over here, the Y has the fewest, 231. Y has the fewest, it is what? 231. And over here, chromosome number 1 has the highest number of genes, that is 2968. Now, scientists have identified about 1.4 billion locations where single base DNA difference is there. That means single nucleotide polymorphism. That means over here, GACC is the thing. Oh, but you have GCCC and some other person has GACC. So this is single nucleotide polymorphism. So scientists have found different sites like that as well. So these are the points that you need to remember. Cool? Now, as I said, I cannot, help you with remembering things, but I can definitely try, and I did try. So now the next question is, describe the lac operon concept of gene regulation. Now this is super easy, okay, and this can come for examination. This is very important. So over here, listen. So this particular thing that you see is a gene. And each gene which is there, it will have its own promoter region. Why? Because it's on the promoter region that the RNA polymerase will come and bind and do the transcription. And after transcription, what will happen? Translation would happen and we will get a protein out of it. So this gene is going to code for some protein. What is the name of this protein? Repressor protein. And what is this gene? Regulatory gene. It is regulating something. Okay, it is regulating something. Just a second. Aju is king. Hi ma'am, I am your old student. I am in RV College of Engineering. Superb. Awesome. Wishing you all the best and I'm super proud of you. Okay, so where were we? Yeah, so this was coding for a protein that is known as repressor protein. Now on the other hand, this is another gene. This has its unique promoter region, and apart from that there is one region which is known as operator region. Now this gene, if you see my dear children, there are different proteins being coded. So Z is coding for another protein, Y is going to code for another protein, and A is going to code for another protein. But all of them are placed together and all of them has a common promoter and an operator region. So since this is a regulatory gene and is coding for repressor protein, you know what it does? It goes and binds on the operator region. Why? Such that the RNA polymerase cannot move forward. So that transcription would not take place. Cool. So this is happening. Now, over here, whenever, this is the case of E. coli. E. coli is having all this thing. So E. coli, when it does not have nutrition in its medium, there is no need of forming all these enzymes out there. But whenever there is nutrition in the medium, what is nutrition? Lactose. Suppose lactose is there in the medium. You know what lactose does? Lactose slowly sneaks into the cell membrane of the E. coli, and then, my dear children, will be binding with this guy that is a repressor protein. Now, this guy would not be free to bind on the operator region. There is no one on the operator region, which is why transcription can happen smoothly and all these enzymes will be formed. Now, what is the role of these enzymes? So, Z codes for beta-galactosidase, which is going to break the lactose. In the nutrient medium, you have the lactose, okay? The lactose is broken down into glucose and galactose, and this glucose is going to give the energy to the E. coli. Fine. And permease is helping in what? Permease is going to increase the permeability of the membrane such that more lactose can get in. Transacetylase is going to deal with everything that happened in between. Whatever conversion, certain things might be there, it deals with that. So transacetylase deals with that. So why do you think it was off over here? Why do you think transcription did not take place over here? Lactose was not there, then what will they break? Which is why there is a off condition. And this was what? Switch on condition. So lac operon, my dear children, works in that way. Always remember that. And the one that induces the transcription is known as inducer, and that is none other than lactose or another form of lactose which is known as allolactose. In exam, is it okay to draw the diagram of lac operon experiment? Yes, definitely you have to draw. Without drawing, do not tell the answer. It's super important. This diagram is super important. Okay? Fine? Now, going ahead. So, switch off mechanism I have explained, and switch on mechanism also I have explained. Z, Y, A are the genes. Zyada, you can remember like that. Zya. Okay. Going ahead to the next question. DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Michelson and Stahl to show that DNA. Can we only draw that in the enzyme? You have to explain that also in few points. Okay. It's not like diagram only would be enough. You would have to explain that a bit. Cool? So, yes. Now, over here, DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Michelson and Stahl to show the DNA replication is semi-conservative. First of all, what do you mean by semi-conservative? So, this is the DNA. And the DNA, when it replicates, the two strands become separate. What do you call the strands? As parental strands, because you got them from the parent DNA, right? So this is the parental strand, and the new strand which is going to forms like this. Agreed? This is known as semi-conservative. Why? Because half is from the parent. So semi-conservation is taking place over here. Now how do you prove it, ma'am? So over here, Michelson and Stahl did an experiment to prove this. They used Escherichia coli, E. coli, my dear children. Remember the name of the bacteria. It was E. coli. And E. coli has its own DNA, right? So E. coli which was there, it was grown in a medium that contained N15 isotope of nitrogen, and DNA has nitrogen in it, you know, nitrogenous basis. So DNA, what it took all the N15 and made the DNA. The DNA would be incorporating this N15 molecule. You have to remember like that. Okay, so it had that in their DNA, and now when this was centrifuged in a medium of cesium chloride, it was found that the band was formed over here. Since it was very heavy, N15 isotope is really heavy isotope, okay? So the band was observed over here. Now the next time, after 20 minutes, they try to, so basically over here they've got this thing, and then they grew the same E. coli in another medium containing N14 isotope. So this time the medium was changed. So now E. coli just had N14 available for making up their DNA. So they made up the DNA with the help of N14. But the problem was that one strand contained N15, the parental one, and the new strand contained what? N14. The same thing happened over here. Now when he centrifuged in the cesium chloride medium, he found the band in between, and this was known as hybrid band. Okay, this was known as hybrid. Why do you think hybrid band was being formed? Because the parental strand was still there. It was not like both of them were N14, right? Now again after 20 minutes, to total of 40 minutes, my dear children, it was noticed that there were 4 strands being formed. Over here, when they separate, this guy goes here and this guy goes here. The new strand is anyway N14, so you get 2 strands of N14 and the parental strand is still there. So here you get the hybrid one and the light chains as well. So there is an extra band that was being formed. So this is how they proved the semi-conservative mode of replication in the case of E. coli. They are of 2 different colors. So you can use black as well as blue pen to draw that, or a pencil and a pen. That would be enough. Okay, a pencil and a pen would be enough to draw this. And then you explain the answer. You do not have to explain the whole thing like this. It's just for you to read and understand. If at all you get whenever you get the PDF in your hand, you can just go through the answers well. Okay? You do not have to write the entire thing. Mention the points out there, whichever is important. Going ahead, we have Oswald, Avery and others who continued Griffith's transforming principle experiment. First of all, you have to go through Griffith's experiment as well. It should not be like you miss the Griffith experiment. That also can come. So definitely do not miss it. Now, the Griffith experiment was done, okay, in that he found that when heat-killed S strains, okay, was mixed with the live R strain, the live R strain was converting into the live S strain. Why was that happening? Where is the PDF? You will get it in the WhatsApp channel. So make sure you go and join the link which is given below in the WhatsApp. After the live ends, I would be uploading it there. Okay? Cool? I hope that works. So, over here, my dear children. So, heat-killed S strains were there. It was taken. And now proteinase was added to this. That means it is going to digest the protein of the heat-killed S strain. When it digests the protein and then the heat-killed S strain was mixed with the R strain, still you saw that the S strain conversion was happening. S strain was still being formed. That means something was getting transformed from this guy to the R strain, converting into S strain. Even if when it was treated with proteinase. The second thing was it was treated with RNase that digest the RNA. Same thing happened, but since again S cells were still appearing, that means digestion didn't take place. Whatever was transforming is still transforming. Now DNA was being put. That means no DNA would be there. The DNA would be digested. After that, when he saw there was no S cell appearing, that means probably it's a DNA that is getting transformed from the heat-killed S cells to the live R cells, transforming it into what? The S cells. I hope you got it. So basically it's a DNase, and this was the experiment done by Oswald, Avery, MacLeod and McCarthy. Hi Navyashri. Okay. Going ahead with the next question. So here explain all of it, and yes. The next question is about Hershey and Chase. So even though these guys did the experiment, that is the Avery, McLeod, or McCarthy did the experiment proving that DNA could be the genetic material, people didn't widely accept it, which is why two other people had to come into picture, that was Hershey as well as Martha Chase. So they did one more experiment to finally prove that DNA is definitely the genetic material, and that was this experiment. So over here what was done? Listen, there was bacteriophage. Remember, bacteriophage is not a bacteria, it's an organism that infects the bacteria, which is E. coli. Now, the bacteriophage's outer covering has protein, okay, and the inside that it has the genetic material. So basically this guy was treated with radioactive S35. So S35, one set of bacteriophage was treated with radioactive S35, and sulfur is a part of protein. So it went ahead and was there in the capsid, in the outer layer of the bacteriophage. Whereas the other group of bacteriophage was treated with radioactive P32. And phosphate, is a part of what? DNA. So over here, the DNA was labeled with radioactive P32. Now both of them were allowed to infect the E. coli. And you know what happened? Basically, we had to find out what other thing that is being passed to the E. coli. Then only we'll get to know what is the genetic material. The genetic material has to be passed on, right? So over here, it was found that, infection was done, and after that blending was done. That means whatever things were left around, my dear children, they were separated with the help of blending. The last thing was centrifugation. Okay, where they were totally separated, and whatever things were there, they will level down. Either they will form pellet or they will form supernatant or things like that. So in this particular condition, they found that no radioactive S35 was detected in the cells. In the cells, no radioactivity was there. That means protein is not getting transferred over here. That was pretty sure. So radioactivity was seen in the supernatant instead. So protein is outside. Protein is not something that is getting transferred. But on the other hand, here, radioactivity was seen in the cells. That means phosphate, which was radioactive, is a part of DNA. So DNA probably could have been transferred. That was being proven like that. Okay. So no radioactivity was detected in the supernatant in this particular case. And here it was. So S35, 5 looks like S, so you can remember S35, and here it's P32. So this was a experiment of Hershey and Chase, who proved that DNA is, yes, the genetic material out there. Cool. Now, explain the process of translation in protein synthesis. This is yet another question. So first of all, the first point is what? Amino acylation or activation of tRNA. So first thing is that the tRNA molecule which is there, okay, on its 3' end, it will have 5' and 3' end. On its 3' end, an amino acid is made to be joined. This process is known as amino acylation or charging of tRNA, which is requiring too much amount of energy out there. And over here, as you can see the picture, this is a ribosome molecule. So first thing that happens is, as I said, amino acylation or charging of tRNA, which needs ATP. And then you have the mRNA, my dear children. Okay, this mRNA which is there, which has to be read, is going to bind with the smaller subunit of the ribosome. And then comes the larger subunit. Wait. So first thing that happens is amino acylation. Second thing, smaller subunit comes and attaches to this. Now the amino acylated tRNA would be coming and attaching over here. Okay, the amino acylated tRNA will coming and be attaching over here. This is the third step. Now the fourth step is that the larger subunit will be coming and attaching over here. Okay. So these are the steps. And then later what will happen, my dear children? The ribosomes will be moving. Again, one tRNA will be coming and joining with one amino acid. And now between the amino acid, there will be peptide bond formation taking place with the help of the enzymes which are there in the ribosome. In the ribosome, there are certain enzymes which helps in the peptide bond formation. In same way, what will happen? When one tRNA comes over here, whatever amino acid is there, it goes that side and this tRNA moves away. So we'll get a strand of polypeptide being formed. Okay, and that is going to form the future protein out there. So that are the steps. So activation of charging of mRNA, then initiation. So always remember, you have to mention this particular part, that is mRNA binds to the smaller subunit and the start codon AUG is recognized by the tRNA containing methionine. So that is the initiated tRNA. So first tRNA always has methionine with it, and AUG would be the start codon in the mRNA. The larger subunit will be binding, forming the initiation complex, and then later on again elongation would be happening. That means tRNA brings amino acid to the ribosomes, matching codons on the mRNA on the with the help of its anticodon, and then peptide formation would be taking place with the help of enzyme that is known as peptidyl transferase, and the same thing goes on. Termination. How does termination happen? There is a stop codon at the end of the mRNA. UAA, UGA, UAG, UGA, UAG, UA. 3 stop codons are there. These are not read by any tRNA. So no tRNA will come and bind over here. Instead of that, what will come and bind over here? A release factor. Release factor comes and binds over here, and that's how we end the process of translation. Cool? So that's how things work over here. So you have to explain line by line like that. And now we will go ahead with the next question, that is question number 19. Children, here, not there. Understanding, not understanding. Is it getting too much? Should we shift to MCQs? If we should shift to MCQs, tell me. We will complete the MCQs first, and that could be better, I think. Or do you want me to continue with this only? Huvama says yes. So we should move to MCQs. Rishi says no, ma'am. So yes and no, yes and no. Let's see the, what do you call, the maximum number of children who tells. So over here, let me see how many. Okay, few 2 marker are there. Let's complete that also. Dhanu says anything is fine. Okay, I see there are some children who are actually coming here to study full things right there. So I'm really proud of you guys. Okay, so what sequences of bases transcribed from the DNA are found both in hnRNA as well as mRNA? What do you think in common both in hnRNA as well as mRNA, my dear children? So when the DNA is transcribed, my dear children, the first thing that you get is hnRNA. It will have 2 type of things. One is exons that will be coding for proteins later on or coding for amino acids later on, and then you have introns in between. So in between the exons you have regions that do not code for anything. You call them as introns. Okay, so this is found in hnRNA. Now what will happen? When you have to make proteins, you do not need these introns. So they have to be removed with the help of a process which is known as splicing. So this hnRNA is going to undergo splicing where the introns are being removed and you just have exons which are going to be joined up together. Okay, now the process formed of hnRNA is known as mRNA. So if at all you are asked this question, what is common here and here? It is the exons, because they are going to code for something. So they are going to be in both the sites. Okay, and always remember, it's not just splicing. There are certain other procedures such as capping. Capping means you're adding what do you call chemical group out there, and if at all you remember the name of the chemical group, write it right now in the comment section. Okay, what is the chemical group that is being added at the 5' end of the transcription, at the 5' end of the hnRNA which is there. Okay, and at the 3' end there is tailing being done where you add so many adenine molecules. Okay, you add so many adenine, and that is known as tailing. So splicing, capping and tailing is being done, and that's how you get the process type of RNA which is known as mRNA. And this mRNA will now finally move out of the nucleus where translation is going to happen. The next process. Okay, the information is going to be translated. So here you are asked what is the sequence of bases transcribed found both in hnRNA and mRNA. It is exons, and you have to explain what are exons. Exons is what? The coding regions. Cool. Now mention the levels where gene expression can be regulated in the eukaryotes. So in eukaryotes, my dear children, so we definitely have different regions where everything is happening. It's not like everything happens in the same region, such as prokaryotes. In prokaryotes, there is no nuclear membrane, nothing like that. So everything happens together, transcription, translation, everything. But here we can regulate to some extent. First thing, how we can regulate? The process of transcription, we can stop it. Okay, so formation of primary transcript, we can stop that. Second thing, processing level, that means we can stop splicing and capping, tailing, all of this thing. Now after that, when mRNA is being formed, we can stop it from going outside. So transport of mRNA can be regulated. And after that, mRNA, when it is outside, you have to do the next thing, that is known as translation. So translation can also be stopped. So there are 4 levels in which gene expression can be regulated in the terms of eukaryotes. Cool? Now, write a note on the role of sigma factor and the rho factor in transcription in prokaryotes. So always remember, in the case of prokaryotes, you need sigma factor for initiating the process of transcription. So RNA polymerase needs sigma factor for initiating the process of transcription, and after it initiates, the sigma factor goes away. And now towards the end, when you have made the RNA and you want to dissociate from the DNA, you have the rho factor, so it starts crying towards the termination. You can remember like that. Okay, so rho factor comes and helps the RNA polymerase to detach from it and remove the RNA as well, the newly formed RNA. So that is the use of the rho factor. Okay, cool. So sigma factor binds to the RNA polymerase, guiding it to the promoter region for transcription initiation. It detaches after initiation, whereas rho factor facilitates termination by binding to RNA and unbinding the DNA-RNA hybrid. Cool. Now next question, what are VNTRs? Mention the steps to detect the VNTRs in identifying criminals in forensic investigation. VNTRs means variable number of tandem repeats. I told you, I will have GACC in my body, and you might have, okay, I will have 8 times in my DNA, but you can have it 10 times. And depending on that, I will be, suppose I did a crime, and when I did a crime, my dear children, there was some blood stains or things like that over there in that particular crime spot. Now that particular DNA was extracted, it was coded, and it was found that there were certain things that was being repeated. Okay, so now I cut my DNA and I got 8 times of this particular thing being repeated. Now they have doubt on me and you. They took both of us, our samples, they cut it with the same enzyme they used for cutting this particular DNA. And then, my dear children, they made the DNA run through an electrophoresis chamber. So in electrophoresis chamber, the DNA will move along with its, its length out there. So if it is big, it will move slow. It is slow. If it is small, it will move fast. So segregation was done, and it was found that, suppose it looked like this. So my DNA strands were like this. Your DNA strands segregated like this. And the possible criminal's DNA strand was also like this, which matches with mine. Get it? That's how you find that this particular person is basically what? The criminal. So there are few things that are being involved. The first thing is isolation of the DNA. Second is digestion of the DNA by the restriction enzyme. That means we are cutting the DNA with the help of some restriction endonucleases, the ones that cuts the DNA. Now after that, you are going to separate the DNA fragments with the help of electrophoresis, where it will be moving from this region to this region. So DNA is negatively charged. DNA is negatively charged, so it will move towards the positive end. Okay, here negative charge is there, here positive thing is there, so it will move towards the positive end along with its length. So the smaller ones can move faster, the larger ones remain slow. Now after, it's not visible over here, so what do? We take a sheet, nitrocellulose sheet, and we transfer whatever DNA is there on this particular sheet. And after that, it is still not visible. Now what do? We basically try to make a complementary base pair of this. So whatever segment which we saw was repeating, we make a complementary segment of this and we radioactively tag it. We tag it with a radioactive item out there. And then now on this sheet which we have, my dear children, we run this particular thing. So it will directly go and bind to certain regions. It will be binding to all the regions. So you also have this, I also have this. So it will go and bind anyways. Here also it will go and bind anyways. And now we will be putting UV radiation to visibly see it. And when we see, we are able to find that the strands, they look exactly similar. So probably this person did the crime. So this is the basis of DNA fingerprinting. And over here, DNA fingerprinting, I told you I am skipping the DNA fingerprinting portion. That question can be coming, right? So this is the same thing. You can answer in 2 different ways. Over here, you do not have to explain much. But there in DNA fingerprinting, you have to elaborate it a bit more. Okay, ma'am, I had Hershey and Chase experiment, 5 marks from the chapter. Yes, biomolecules are from the chapter molecular basis of inheritance, right? Yes. So yes, my dear children, we have already studied that. So you can expect all these questions coming in. I am telling you, these are some things that are repeated most of the times. So please have that in your head and try to solve it. So isolation of DNA, then you cut the DNA, then you separate the DNA, then you transfer it into a synthetic medium, and then you hybridize using VNTR props, the props that you have made, and then you detect it with the help of UV radiation. Okay? Cool? So Kumuta, thanks for coming and telling that yes, it was there, so that all the other children are also aware that yeah, these type of questions can be repeated. Just give me a second, okay? Hello? Sir? Sir, you can keep it outside. Yeah. Sir, you can keep it in the lower, ground floor. At the first house. There are two buildings. The first house in the 7th cross is the house. Okay, and the ground floor there is security guard you can give to them.
This apartment name Sri Balaji Residency 349 7th cross Kerala. Space. Okay.
I think it's not 349, it's the opposite, the first one. Yeah, the first one. First one in the 7th cross, first building. Okay? Yeah. Building number I don't remember as of now. Okay. Thank you. Oh my God. Okay. Sorry, children. Okay, so we are here. So write the differences between euchromatin and heterochromatin. That is the next question that we have. So what is the difference between euchromatin and heterochromatin? So DNA which is there, it is compacted, okay, to form a chromatin structure. What is chromatin? So over here you have a nucleosome. That is basically it is made up of histone. How many histone proteins? 8 histone proteins comes together. And around it wraps our DNA. How many times? Twice. Okay. And then it goes around, right? Suppose over here, the nucleosomes are very close to each other. You call this structure as heterochromatin. It's very compact. And over here, suppose they are far away from each other. There is some distance. You call this as euchromatin. So as you can see, it is loosely packed and here it will be tightly packed. And since it is loosely packed, it will stain lightly. Over here, it will stain darkly or intensely. And since the DNA is still there, transcription can happen. The RNA polymerase can come and do the transcription easily. So this is transcriptionally active and this is not transcriptionally active. So these are the three things that you have to remember. So in a typical nucleus, some loosely coiled regions of chromatin with light stained and transcriptionally active is there. That is known as euchromatin. The chromatin that is more densely packed and stains dark and are transcriptionally inactive is known as heterochromatin. Okay, so these are the few points that you can definitely write. Fine? Cool. Now the next thing, name the virus used in the Hershey and Chase experiment. What is the name of the virus that we used in the Hershey and Chase experiment? It was bacteriophage. I told you, is the one that infects the bacteria. It itself is not the bacteria. So please remember that as well. Cool. Now going ahead to the next question. Mention the criteria for a molecule that acts as a genetic material. So what is the criteria for a molecule to act like a genetic material? It should be able to replicate, my dear children. Right? If it is not replicating, can it give to the other? If a cell divides, it has to replicate, then only it will be passed on to the generations, right? Now it should be chemically and structurally very, very stable. If it is not stable, what will happen? It can react to anything and they will be not exactly like this. And it should provide the scope for slow changes. So it's not like no changes should be happening. It's not like that. Changes should be happening. That's how evolution can happen. That's how better things can come up. But there should be slow changes. Now it should be able to express itself in the form of Mendelian character. That means whatever DNA is there, it should be having genes and the genes should be coding for proteins. That is what you mean by it should be able to express itself in the form of Mendelian character. Okay. So these are the things that you need to write, and this is the reason why RNA is not a good genetic material. Why? Because RNA is not chemically stable. You know, in the case of RNA, on the second carbon it has an OH group which makes it highly unstable. Okay, which makes it highly unstable out there. I hope that is also clear. And now we have the next chapter. Again, as I said, Do we need to go ahead with the MCQs, or should we target the 5 marker, 3 marker, or whatever questions have come from the chapter? What do you think? Depending on it, we will be going ahead. Gayatri, Griffith's experiment is important. Yes, Griffith's experiment is also important for a 5 marker question, okay? So we will be going ahead with MCQ is what you're saying. So these are the MCQs that we solved yesterday. So over here, the first thing, a chemical structure is given below. Identify its plant source. What is this particular thing? What is this particular thing? The structure is of C. Okay, so Prakriti, we learned it yesterday. The answer is C. Papaver somniferum. Right? So chemical structure is given below. Identify its plant source. This is what? It is an opioid. What is this? This is actually morphine. It looks like O, right? So opioid. And morphine, where do we get it from? You get it from Papaver somniferum. So your answer for this question becomes C. Okay? Next, one of the following is not the causal organism for ringworm. Which is the one that does not cause ringworm disease? Any guesses out there? We discussed this yesterday. Do you have any guesses? Just give me a second, okay? Children, just give me a second, a quick second. Okay. I'm so sorry, I was getting a repeated call. So everyone is saying D, Prakriti says D, Gayatri also says D. The answer is D, microsporum. So all the other thing, microsporum, trichophyton, epidermophyton, they cause ringworm disease. They are all fungi, my dear children. They are group of fungus. So macrosporum is the odd one out. Now going ahead with the next question out there, which is this. An intestinal parasite which causes blockage of the intestinal passage and whose eggs are excreted along with the feces of the infected person is what? So there is a parasite that causes blockage of the intestine, and not just that, it is found, the eggs of this particular parasite is found in the fecus of the organism. That means the stool of the human being out there. So what do you think is this particular parasite? Ascaris, yes, the answer would be B, and it causes ascariasis and it causes internal bleeding as well. Okay, it can later cause internal bleeding as well. So this would be your answer for the question. Now, going ahead with the next question out there. Which of the following is affected by the infection of Wuchereria bancrofti? Which of the following is affected by the infection of Wuchereria bancrofti? Try this. Lymphatic vessels, respiratory system, nervous system, or blood circulation. Wuchereria bancrofti. Okay, Prakriti says A. Gayatri also says A. Any other guesses? So the people who are watching it, it is lymphatic vessels which are affected. So as I told you yesterday, the lymphatic vessels which are there, they drain out the extra fluid which is there in between the cells. So if at all lymphatic vessels are blocked, the extra fluid remains to be there itself, and it can cause swelling, which is why it looks like an elephant's leg. Elephantiasis is being caused because of this particular thing. And now, can you guess one more very important thing? If at all you know, tell me the answer. What is that mosquito that spreads Wuchereria bancrofti, or basically spreads the disease that is known as filariasis or elephantiasis? So you know malaria is caused by Anopheles mosquito. Dengue is caused by Aedes mosquito. What about this particular disease? How is it caused? What is the causative organism for this? Any guesses? What is the one that spreads? What spreads filariasis? Which mosquito? Culex mosquito. It is Culex mosquito. Do not forget it, okay? Fine. Now going ahead with not Anopheles mosquito. Listen, Anopheles mosquito is for malaria. Culex is for this particular thing, and you have one more thing which is known as, what do you call, this thing, chikungunya as well as dengue, that is caused by Aedes mosquito. Cool. Next thing, which of the following pairs correctly matches a disease and a pathogen causing it? Which of the following pairs correctly matches a disease and a pathogen causing it? Any guesses over here? Correctly matches a disease and the one that causes it. No guesses? So the answer for this question is typhoid. Pneumonia is caused by Haemophilus influenzae, not pneumonia. Malaria is caused by Plasmodium. Okay, and ringworm is caused by Microsporum, Epidermophytum, Trichophyton, all of them. So Harshita, you were right. The answer is A. Even Gayatri is right. The answer is A. Cool? Fine. Now going ahead with the next thing, that is hemozoin is what? What is hemozoin? Prakriti says, isn't pneumonia caused by Streptococcus pneumoniae? So there are two causative agents for pneumonia. One is Haemophilus influenzae and the second one is Streptococcus pneumoniae. Okay, there are two causative organisms. Now over here you have hemozoin. The next question is there. Tell me the answer for this. Children who are watching this video, chalo. We have to wrap this session very quick, so give your answers really fast. Now I want full efforts out here because we are solving just MCQs. No 5 marker, 3 marker, right? So complete efforts over here. Tell me the answer for this question. What is hemozoin? It is, Prakriti says C, Gayatri says B. Let's see who is correct. So C is what? Toxin released from Plasmodium infected cells, and B is what? Toxin released from Streptococcus infected cells. So hemozoin basically is a toxin which is being released when a cell is infected from Plasmodium, because as I told you yesterday, in RBC there is hemoglobin and the Plasmodium comes to eat this hemoglobin, right? And it breaks them down into heme and globin. This heme is toxic for the Plasmodium, which is why it converts into hemozoin, which is actually toxic for us. So your answer for this question would be C. Okay, this is C. So here I have mentioned the life cycle of Plasmodium. You can just go through it. The stage in which it infects us is known as sporozoite. It is found in the salivary gland of the Anopheles mosquito. And then basically inside our body it keeps on doing asexual reproduction, no sexual reproduction. Okay, and at the end it forms a stage which is known as gametocyte stage. What stage? Gametocyte stage, my dear children. And basically the mosquito will come and drink our blood. It takes up the gametocytes, and in the mosquito's gut the gametocytes form gametes, and the gametes fuses to form next form of Plasmodium. So that's how it goes. So always remember, it infects our body in the form of sporozoite stage. Where do you think the gametocytes are formed? It's another stage. Okay, it is formed in the RBC, and sexual reproduction does not take place in our body. Cool? So over here we have the question. During the life cycle of Plasmodium, gametocytes develop in. Where do you think the gametocytes develop in? In the life cycle of Plasmodium, where do you think the gametocytes would be developing in? Human RBC, human liver, salivary glands of mosquito, gut of the mosquito. 7th question, answer. Children. Come on. During the life cycle of Plasmodium, gametocytes develop in. Harshita says the answer is D. As in, they are asking you, where are the gametocytes formed? Not the question is not like gametocytes are going to divide where. That's not the question. They are asking you, where are the gametocytes formed in? That's the thing. So now think the answer. Where are the gametocytes formed in the what do you call, in the plasmodium, my dear children? It would be human RBC. I told you, in the RBC, the gametocytes would be formed. Later on, these gametocytes are going to give rise to gametes in the mosquito's gut. So that is the thing. So over here, yes, Harshita. Now it's correct. Your answer would be A. Okay, so I have marked it wrongly as liver. It is A. It's human RBC. Cool. I know the question is kind of a bit confusing, but yeah. Next question. The human immunodeficiency virus primarily attacks and destroys what? What does it attack and destroys? Come on, dear children. HIV virus, which is the cell that is being attacked by HIV virus? So usually it is macrophages. Macrophages is the factory of HIV. Macrophage is where this HIV viruses will come and proliferate. That means increase the number. Okay, but you are being asked about which are the cells which are mostly affected by this particular thing. So Rocky is also here. Prajwal is here. There are so many new people. Good. Who is there? Oh my God, lot of children are coming up. So you are saying B. The answer is exactly B, T helper lymphocytes. To T helper lymphocytes help everyone for immunity. Now when these guys are targeted, immunity would be lost. Immunity will be greatly reduced. Okay. So if at all you are asked about what is the factory of HIV, it is macrophages. If at all you are asked what are the cells that are being affected mostly, it is T helper lymphocytes. So you all are correct. And now going ahead, the substance produced by a cell in viral infection that can protect the other cells from further infection is known as. So there is a substance that is produced from a cell in a viral infection. Suppose this particular cell is being infected by a virus. Okay. And now it releases something and says the other cell that, hey, you have to be safe. What is that particular thing that has been released over here? Rocky says the answer is C. Prakriti says the answer is C. Okay. Anyone to challenge them? Anyone to challenge them? Rocky says C. Prakriti says C. Hovama also says C, so I think no one is there to challenge them. They are absolutely correct. It is interferons. It is interferons. Okay. Now, I want to ask you a question which only those children can tell who basically have read the NCERT really well. One type of interferons are given to cancer patients for modifying their immunity. They are immunity modifiers. What is that type of interferons known as? One type of interferons are given to the cancer patients for boosting up their immunity, for modifying the immunity. What is that type of interferon? So those of you who read your NCERT really well would be able to answer this question, and those of you who have not read won't be able to answer this question. Any guesses? Any guesses out there? Alpha interferons. Yes, it is what? Alpha. You call it alpha interferon. So that is given as biological modifier for immunity. Okay, good, Prakriti. Awesome. So Prakriti has read the NCERT really well. Now going ahead with the next question. Antibodies present in the colostrum which protects the newborn from certain diseases is. Remember the small details which is given in the NCERT, you have to read it. Okay. Now antibodies which are present in the colostrum which protects the newborn from the certain diseases is known as. Rocky says the answer is B. Is it B? So what is colostrum? The first milk which is released by the mother after the process of parturition. Harshita also says is B. Prakriti also says is B, and the answer is actually B. It's IgA. AA stands for Amma. You can remember like that. I have told you yesterday also. The first milk released by Amma, it has IgA which provides immunity to the children. It is yellow. The colostrum is yellowish milk. Okay. Now next. Okay, what type of immunity is it? When you are giving ready-made antibodies, what do you call this type of immunity as? Very important question. What do you call this type of immunity as? And you try to solve this question as well. Question number 11. Vaccines work by. How do vaccines work? What do they do? Stimulating the production of antibodies, killing the pathogen directly, reducing inflammation, blocking the entry of pathogens into the body. Yes, Rocky, it is passive immunity because you're giving ready-made antibodies. Absolutely correct. Good going. So vaccines work by what? How do they work? What is the action? So when we had COVID era going on, my dear children, at that point we were all given vaccines. Why? So these were, in vaccine we were giving the spike protein which was there on the coronavirus. No, it has some spike protein, so we were giving a part of the that particular thing or something made from that. It was given to us as that our body knows something like this can attack us in future and we have to release more antibodies. So for initial response, for primary response, we were given that vaccines. So it is for stimulating the production of antibodies. Okay, so A is your answer. So Prakriti gave me the correct answer. Rest all of you didn't try. So children, you have to try, then only you will be getting your answers correct. Okay. Then only you will be learning from mistakes. It's not like you always know the correct answer. That need not be true, right? Fine. Next question out there. Which of the following cells actively participate during allergy? Which cells do you think actively participates during allergy? B lymphocytes, liver cell, mast cells or red blood cells? C. Mast cells. Is it mast cells? Yes, it is mast cells, because mast cells, my dear children, would be having antibody E on their surface, and you know what? Antibody E would be binding onto the dust and mites and whatever causes allergy, and now signals the mast cells to release two things. One is histamine and the other one is serotonin. Okay, and the histamine will be leading towards dilation of the blood vessels, so that more cells can come in. When the vessels are dilated, more cells would be coming in, and that's when you get a hyperactivity thing going on there, exaggerated response. Okay, so the answer for this question is C. Yes, Huvamma as well as M. Mr. Shankar, you are correct. Now, over here I have the 5 markers. I'll quickly show you. You can be asked about the lymphoid organs, each of them. Go through it clearly once. Okay, the primary lymphoid organs you should know, it's bone marrow as well as thymus. Talk about it. And now explain the different techniques which helps in cancer detection and diagnosis. Go through it. Really important question. All the cancer features of cancerous cells, detection methods, what are the causative agents of cancer, all of it is important. Over here the drugs are given, so there is a question where the label of the drug is being missed and you have to tell what is the drug. So used by doctors as sedative and painkiller. Sedative and painkiller is morphine, helps the patient to cope up with insomnia, that means lack of sleep. It will be barbiturates, amphetamines and benzodiazepines. Increases blood pressure and heart rate of the consumer, it will be nicotine. Okay, it increases the BP. The ones who smoke will have high BP. Now, acts as depressant and slow down the body function. It is what? Heroin, the opioid, and affects the cardiovascular system. So, there is only one drug that according to NCERT affects the cardiovascular system, and that has to be cannabinoid. Okay, so blood pressure with nicotine and cardiovascular system with this. CNS is affected by opioids as well as coca alkaloids. So, these type of questions can be coming up there. Okay, schematic representation of replication of retrovirus, super important. Talk about all the steps nicely. Okay. Maths class will be there tomorrow, Santosh. It will be there tomorrow, okay. 3-marker question again. What is the name of the drug? Source plant is given, Papaver somniferum. Anticipating that it is a depressant, so it has to be heroin. Okay, cannabinoid, my dear children, the source plant of the cannabinoid is what? Cannabis sativa. It has effect on the cardiovascular system. Cocaine is the name of the drug. Its source plant is what? Erythroxylum coca. And the nature of it, what does it do? Cocaine, it affects the central nervous system. So these are the things that you should definitely remember. So 3 properties of cancerous cells: they are uncontrolled growth, they metastasis, that means they move around through body, they have loss of contact inhibition, so they don't stop while dividing, all these things. Differentiate between active and passive immunity. Talk about allergy. This can be a question. Go through all these things. Super important. Types of carcinogens, the one that causes cancer. Okay. And the structure of antibody. This particular thing is super important from this particular chapter. So go through the diagram. 2 methods of HIV transmission. Talk about them. And yeah. Now we are coming to reproductive health. These are the topics that you should not be missing. Okay. Amniocentesis, the genetic disorders that can be found with the help of amniocentesis, such as hemophilia, Down syndrome, all of it, go through it once. Amniocentesis was banned because it increased the female feticide. The female children were killed. Infant and maternal mortality rate, go through it. Contraceptive methods, especially IUDs and oral pills, both of them are most widely accepted things are these two. And the ones that is perfect for spacing children is IUD. Please make sure you go through IUDs really well. MTP, when is it safe? Until 12 weeks it's safe, but you can perform it up to 24 weeks, and the act which was there of MTP, go through that as well. And now STDs, the non-curable ones, super important. There are 3 of them. Everyone starts with H: hepatitis B, herpes, as well as HIV/AIDS. Now ART, ZIFT, GIFT, and everything you should know. ART means assisted reproductive technologies. So all these things are really important. Prakriti, yes, first trimester. Good. So I'm really proud of those children who come back over here, tell me the answer, because I clearly see that they are working really hard. Super proud. Okay, so these are the topics that you need to cover, and these are the questions that you need to cover for definitely from this particular chapter. I am not going ahead with a lot of explanation for this. And now you have evolution chapter. Again, these are the set of questions that you need to practice. Salient features of human ancestors, all of them. Detailed note of adaptive radiation, give example of finches. Help with the label, neat diagram, Stanley Miller's experiment, and what were the things that were used. Okay, what answer for this question? Fusion of gametes and zygote formation does not occur within the body of female. Fusion of gametes and zygote formation does not occur within the body of a female. It can be ICSI, intracytoplasmic sperm injection. Or you can say IVF. Or IVF, if you have an option of IVF, in vitro fertilization, that would be your answer for this question. Okay? Or if you have an option of ICSI, ICSI, that can be the answer. So depending on the options, the answer varies. ICSI is basically what? In the cytoplasm of the egg, you are inserting the sperm. Okay, and that's how fertilization will be happening. That is happening outside the human body itself, and that's how you get the zygote. So ICSI, everything is a part of IVF, in vitro fertilization. That is, in vitro means in the lab fertilization is happening. Okay, Rocky? Now over here, with the help of neat label diagram, yeah, you have to tell the name of the gases involved: methane, ammonia, H2O, as well as H2. Now state Hardy-Weinberg principle. Explain any 4 factors affecting Hardy-Weinberg principle. You have to tell that. Okay. So you can say, Rocky, you can give your answer like, so whenever fertilization is not happening inside the body of the female, it will be known as in vitro fertilization. That means it is happening in the lab. Now in the lab, the method that is used is known as ICSI, ICSI, intracytoplasmic sperm injection. So you can elaborate your answer in this way, so that no ways you would be in the safe zone. I mean, in every ways you would be in the safe zone. So IVF also you have also mentioned about ICSI. So you're elaborating your answer in that sense. Okay. Ma'am, how many types are there in ART? In ART, assisted reproductive technology, you have ZIFT, zygote intro-fallopian transfer, where what do? You get the zygote by in vitro fertilization, and you put it in the fallopian tube. You can either put the zygote, you can put the embryo up to 8 blastomeres stage, up to 8 blastomeres, not beyond that. If at all you are putting it, that will be another stage. It is another program that is known as intrauterine transfer, where we are putting in the uterus. So blastomeres, if at all it's embryo which is more than 8 blastomeres, you're putting in the uterus, it is known as intrauterine transfer. And then you have gift, where suppose the woman is not able to produce viable gametes, so you're giving the gametes, but she has all suitable environment for nurturing the kid. So that is known as gift, gamete intrafallopian transfer. Okay, and then you have artificial insemination. Suppose the male is not able to ejaculate, or the number of sperms are not enough, or the erection is not happening properly. You do artificial insemination, where you are artificially giving the sperms inside the female body. Now, if at all you are specifically putting it directly inside the uterus, you call this intrauterine insemination. If you are putting in this in the cervical region and things like that, in common you call it just like artificial insemination. So these are the things that you need to remember. Okay. So ZIFT, gift, artificial insemination, 3 are there. In artificial insemination, you have one more thing that is known as as I said, intrauterine insemination. So under AI, you have one more thing. That's all. And as also there is intrauterine transfer. So there are 4. There are 4. ma'am, is just 2 marker and 1 marks for this question, and ma'am, I have wrote ICSI, will I get marks or no? So you have to specifically talk about IVF first, because it's clearly mentioned it is happening outside the female body, and then you have to talk about ICSI. So IVF would be more correct if you say that, and just to support the answer you are telling ICSI. Okay? So depending on the teacher you may get marks, but it would have been more correct if you have mentioned both. Fine. So Hardy-Weinberg principle, my dear children, you have to tell about the formula P square plus 2PQ plus Q square is equals to 1, and mention the definition of it, explain the factors affecting it. It can be gene flow, and then what is it? Gene flow, and then genetic recombination, mutation, genetic drift, all these things. Cool. Now potato tubers and sweet potato tubers are a result of convergent evolution, so you have to tell that both of them are arising from different structures, but since they are in the same habitat, they are responsible for storage. So potato tuber is from stem and sweet potato tuber is from root. So it's a root modification, it's a stem modification. Okay. So basically Shashi, the charge of DNA, you say it's negative, it's negatively charged. Okay, whereas because it has phosphate group, and now when you talk about histone is made up of amino acids. Amino acids are both negative and positive, but majorly histone protein is made up of 2 positive amino acids, that is lysine as well as arginine. So lysine and arginine, which is why the DNA is able to wrap around it. So positive, negative, they will attract. Okay. Now write a short note on Neanderthal man. You have to talk about them, that they were the first human beings who basically started covering their body and also burying their dead. Their brain capacity or cranial capacity was 1400 cc, all this thing, and they arose from what? Homo erectus. Okay, they came from Homo erectus. So you have to talk about all the small details out there. Then mention the evolutionary significance of the following organisms, that is shrews and lobfins. So the bridge between reptiles to mammals is shrews, and what is lobfins? The bridge between fishes and amphibians. So fishes and amphibians, the bridge between that is lobfins. Cool. Now next, what are the analogous organs? Given examples. So in convergent evolution, the things which have no similarity anatomically, my dear children, they are analogous organs. Okay, and they do convergent evolution to be same because they are in the same habitat. So give an example, you can just give this example of this particular thing. So you remember like this, AC and HD. Homologous organs are in divergent evolution, analogous organs are in convergent evolution. Okay, now enumerate on convergent and divergent evolution and give examples. So for divergent evolution, you can give the examples of hands and every limbs of what do you call human beings, whales, and what do you call that thing? Bat. Okay, bat used for flying, whales used for swimming, and we use for eating. Okay, so basically you can give examples of divergent evolution with the help of that, and convergent, as I said, you can give this example, or you can give example of butterfly and bird. Both of them have wings, but do you think they are anatomically same? No. Butterfly's wing we can do like this. Bird's wing, can we do like that? No, you should not be doing like that. Okay, so anatomically they are different, but since they are in the same habitat, they are converging to a point. So that is an example of convergent evolution. Nazia, my dear child, tell me, what is the explanation that you need? Alan is back, and the animals have no fingers on the limbs. Yes. Yeah. So guys, Nazia, if you have any doubt, dear, if I went fast, you can ask me right now. I can explain it to you once more. So always remember, divergent means they were having some common ancestor, but they are diverging. Why? Because they moved into different habitats, and for better adaptation, they have formed different structures out of it. But anatomically, the structure is same. So four limbs of whale and us as well as bat is made up of the same thing: radius, ulna, humerus, carpal, metacarpals, everything. But now they are doing different things. That is divergent evolution. Convergent means you are converging to a point. Why? Because you are in the same habitat. So even though you do not have any common factors, you are coming together, living in the same habitat, which is why you will be having some common factors arising. But anatomically, you are not going to be the same. Okay, so these are the things that you need to cover. And with that, my dear children, I would end the live. Okay, is there any doubt that you have? You can ask. And also, as I said, so there were so many, so I have made a video on the short questions that you can definitely practice. It's there. Please go and watch that video. It's going to be helpful for you. I have told you the concepts and I have recorded the one-shot videos. If at all you face any difficulty understanding a concept, you can watch the one-shot video, put it in 2x speed if at all you need, and then go ahead, solve the questions. Okay? So I think I was out to eat the lunch, so I went. Okay, Ella, no problem. Okay, so dear children, so now we would be taking the live after some days. So for biology, I think all the lives have been officially done, but please stay in contact through the videos that we make. Okay, so be there, and wishing you all the best if at all you are someone who has your examination tomorrow. Okay, shall we? Huvamma, you're most welcome, dear, and please go through all the questions. Take care of yourselves. Bye-bye. Yes, Alan says all the best to the juniors. So I hope that wish has been taken. So bye-bye, dear children. Take care. Prakriti, thank you so much, dear. Lots of love. Take care of yourselves, and yeah.